Okay, so we are on, and welcome to everybody. This is our seventh webinar for this new cycle, and I'm very happy with today's speaker, which is a longtime friend, other than a very good, excellent researcher. Today we have Salvador Caballero, which I met here in Europe, but now is a postdoc fellow at the University of Saskatchewan. And today we're talking about breeding for synergy. So please, Salvador, the microphone is yours.
Okay, thank you, Nelson, for the charming introduction. And I'm going to share my slides. Give me a second. Okay. Can you see the presentation mode? Okay. So let's start. Good morning, everybody, and thank you very much for the to the International Legumes Society for the invitation to present this work in today's webinar. Actually, it is a pleasure to share some of the work we are developing within this exciting project. And today I will talk about breeding for synergy in crop rotations. More specifically, I will focus on how we are trying to estimate a legacy value in a lentil-wheat rotation trial, and how this value could help us breed crops that perform better, not only as individual species, but also as partners in rotation systems. Okay, now. So let's start from the beginning. Pulses are important part of global agriculture because they are essential in human diets in many regions of the world, and they are also key components of animal feed systems. This figure shows the most widely produced pulse crops globally using recent FAO data, and beans, chickpeas, peas, cowpeas, and lentils are among the most important pulse crops worldwide. I would like to highlight two of them in particular, peas and lentils, because for both crops, Canada is one of the leading producers in the world, and their export contributes significantly to the Canadian agricultural economy. It's therefore not surprising that over the years, the yield of both crops in Canada has increased. And this improvement is related to better crop management, but especially to the development of high-yielding cultivars that are well adapted to high latitudes and longer photoperiods. So this increase in lentil and pea productivity in Canada is not random. It is the result of a strong scientific and breeding institutions that have supported pulse crop improvement for many decades. And a very important part of this work actually is happening here in Saskatchewan, because this province is where most Canadian lentils and peas are grown, and it is also where the University of Saskatchewan and the Crop Development Center, or the CDC, have played a major role in developing the cultivars grown by farmers. Actually, since the 1970s, the CDC has released more than 500 crops varieties in general, and today around more than 40% of the crop area in Western Canada is planted with these CDC varieties. In the case of the lentil, almost the entire Canadian lentil area is planted with varieties developed by the CDC. And this breeding effort has a direct economic impact because for every dollar invested in pool research and breeding, growers receive a strong return estimated at around $44. And this shows how important breeding is, not only scientifically, but also economically. Now, after highlighting the importance of pulse breeding here in Canada, I would like to move to the main agricultural problem that this project aimed to address. Because modern agricultures depend heavily on nitrogen fertilizers to maintain high yields, especially in cereals crops such as wheat. However, the production and use of these fertilizers is not fully sustainable. So because producing these fertilizers requires a large amount of energy, and also after applications, as we can see in this figure, only a part of the nitrogen is uptaken by the crop. The rest of the nitrogen can be lost from the system. Some nitrogen can leach into groundwater, and some can be transformed by soil microorganisms into other nitrogen compounds, including nitrous oxide. And this gas is important because it's a much more powerful greenhouse gas than CO2. So as we can see in this graph, agriculture is the main source of nitrous oxide emissions, both globally and also in Canada. And there are two main ways to reduce these emissions in agricultural systems. One approach is to target these microorganisms responsible for these nitrogen transformations. And another approach would be to reduce the amount of synthetic nitrogen fertilizer applied in the first place. Both approaches are considered within this project, but in this presentation I will focus mainly on the second one, reducing the need for nitrogen fertilizer by improving crop rotations. So let me first summarize how lentil can benefit the following crop in a rotation. As most of you know, lentils can fix atmospheric nitrogen through symbiosis with rhizobia in their root nodules. And part of this nitrogen is used by the lentil plant for its own growth and development. Some of the nitrogen incorporated by the plant leaves the system when the lentil seeds are harvested, because a large fraction of nitrogen is stored in this seed protein. However, another part of this nitrogen remains in the rotation system. It can remain in the soil or in the plant residues after the harvest. So the following crop, in this case wheat, can take up the mineral nitrogen released from the composing lentil residues or from the nitrogen that was fixed and remained in the soil in the first place. So if this is how the system works, and if we want to improve both crops together, the main questions become which lentil genotypes live better or worse conditions for the following wheat crop, which wheat genotypes are better following crops after lentils, and what is the role of the living microorganisms in the soil in this movement of nitrogen from one crop to the next. So if we wanted to answer each of these questions separately, we could use we could use the current crop breeding framework. In this modern breeding approach, we combine genotypic information, phenotypic data, and environmental information to improve specific traits in a given species and environment. As I said, this is the current scheme in plant breeding, and it is also the basis of many modern breeding approaches that include also genomic selection. This approach has been very successful, and it is still the standard method used in many breeding pipelines, with the limitation that we can improve one crop at a time. Actually, in the lentil breeding program here in Saskatchewan, this general framework has already been successfully applied. And for example, it helped to develop lentil lines adapted to long photoperiods with favorable consequences in yield with longer vegetative period. So as we can see here in these plots, the newer lines developed by the CDC not only have longer vegetative periods, but also they yield more. However, as I said, this approach has an important limitation because it usually improves one crop at a time. And agriculture is more complex than that because a farmer may grow one crop in 1 year and a different crop in the following year. But very often, breeding programs provide solutions just for each crop separately without considering the full rotation system. So if we want to expand this breeding triangle, we can place another crop next to it. In this case, we place wheat next to lentil. And in a crop rotation system, the environment is not completely independent between years because the soil is the common link that connects both crops. So the soil carries nutrients, residues, microorganisms, and other biological and chemical signal from one crop to the next. From this idea, we can define a rotation effect, and this rotation effect may be partially determined by the interaction between the genotypes of the two different crops grown in this sequence. So in other words, the performance of wheat may depend not only of the wheat genotype and the environment, but also on the lentil genotype that was grown before it. So the key question now is whether this rotation effect exists, and if it does, whether there is enough genetic variation to use it in breeding. If the answer is yes, then we may be able to breed the crops specifically for better performance in rotation systems. And this is the main idea behind the field experiment I'm going to talk about. So let me take a look at the experimental design. In our field experiment, we wanted to evaluate genotype by genotype interactions between 100 lentil genotypes and 100 wheat genotypes. In theory, this gives 10,000 possible lentil-wheat combinations, so in this case, size really matters. But of course, evaluating all 10,000 combinations would be extremely difficult. Instead, our trial was designed to evaluate 1,000 of those 10,000 possible combinations. So as you can imagine, this is a very large experiment. And when we include the replicates, locations, years, and the fact that we are working with two different crops, by the end of the field trial, we will have evaluated more than 17,000 plots. So as I'm showing you in the left here, in year one, we planted 100 lentil genotypes. Each one replicated 10 times. And then in year two, wheat was planted on exactly the same plots. Again, using 100 wheat genotypes replicated 10 times. And this creates our 1,000 lentil wheat combinations with each combination replicated twice at each site. I know that this can be kind of confusing, so to understand the design more clearly, let's follow these plots I am highlighting here as an example. And we can represent these lentil wheat combinations as a table. On one axis, we have the 100 lentil genotypes, and on the other axis, we have the 100 wheat genotypes. This field experiment is not only about finding the best lentil wheat combinations. If we place a trait of interest in each cell of this table, for example, let's say the yield in year two, so the wheat yield in year two, we can use the concept of general combining ability. And this would allow us to estimate a phenotype for each lentil genotype that would tell us how well it supported the wheat yield in the following crop in the rotation. So in other words, it would help us to identify the best givers, and later we can use those values to map the regions on the lentil genome associated with a better rotational effect. We can also do the same from the wheat perspective, where we can also identify wheat genotypes that perform better after the lentils. These would be the best takers, and eventually map also genomic regions in the wheat associated with a better response to the previous crop. Now, let's talk a little bit about the timeline of the first rotation cycle. So let me go through step by step the timeline of this first rotation cycle, which took place between 2024 and 2025. We started in spring 2024 by collecting soil samples before lentil sowing. The goal of this was to estimate the baseline level of nitrogen in the field at the beginning of the lentil season. During the lentil growing season, we collected different types of phenotypic data, including phenology-related traits, agronomic traits, and more. We also collected spectral images weekly using the UAVs, and at harvest, of course, we measured the yield. At the time of harvest, also we collected soil microbiome samples to evaluate whether the lentil genotypes are influencing the diversity and composition of the soil microbial communities. And during the fall, we analyzed the lentil residue samples to quantify nitrogen and carbon content in the straw as well as in the seeds. Around 1 month after harvest, we collected also additional soil samples to evaluate whether the nitrogen coming from the lentil residue had started to mineralize and become available for the following crop. During the winter, actually, there's not much to say about the winter field activities because in this part of the world, winter is cold and quite harsh, so the outdoor work is very limited. However, in this period, it's very important for us because it's when we use this time for processing the thousands of samples collected during the season and for starting also cleaning, organizing, and preparing the data. So the second part of the first rotation cycle started with the wheat sowing in spring 2025, following the same plot configuration described earlier. And for those wondering how we make sure that the wheat is planted exactly on the same plots as the previous lentils, well, the seeders use a GPS-guided system that allow us to match the plot's position very accurately from one year to the next one. And at this stage, we also measured the baseline soil nutrients again. And during the wheat season, we collected agronomic and phenology traits again. We also obtained the spectral indices through the UAVs. We also sampled the soil microbiome to test whether the lentil-associated microbiome influenced the wheat-associated microbiome in the following year. And of course, we also measured nitrogen and carbon content in both wheat straw and grain seed. So before moving to the results, I would like to emphasize that the large amount of data we generated by this field experiment, and because of this, it has been essential for us to organize the information into different data layers to keep clear records of the methods we have been used and to make regular backups of all the data generated. So the genetic databases and web platforms developed by the NoPulse team have been fundamental for this work, and without that support, managing this experiment would have been very much chaotic. Also, we are documenting protocols in protocols.io, where we already have a couple of public protocols available. And the data analysis pipelines will also be shared through GitHub as the project advances. So let's move to the actual results, and I would like to show you first that even though the sites where the experiment was replicated were actually relatively close to each other, around 22 kilometers apart, delaying the sowing by a couple of weeks between sites created clear environmental differences. So the two sites in 2025, you can see in the PCA, were more similar to each other. However, when we compare them with the two environments in 2024, the differences are much larger. In this analysis, the site called Hunter in 2025 and the site called Clavet in 2024 appear as the most contrasting environments. And this is very important to us, these environmental differences, because these differences may influence not only the lentil and wheat performance, but also the rotational or the legacy effect that we are trying to estimate. So, as expected, this environmental and the genetic variation that we have in the collections affected the traits we measured in the lentils. So, this is the variability in the lentils, and was also different in each environment. And this can help us to understand whether the legacy effect or the rotational value is influenced by the genotype and also by the G by E interaction. For the lentil trials, here are the box plots, the histogram, sorry. I would like to highlight the differences observed in yield at the right and also the seed protein content. Both, it's important to say that both traits showed significant differences across sites and years, which give us useful information to explore possible drivers of this legacy effect. Let's move to the wheat. For the wheat, the environmental differences were less strong, mainly because the wheat was grown in 2025, and as you can see, as you saw in the previous PCA, the environments in 2025 were more similar. Even though the 100 wheat lines showed clear variability for all the measured traits, and we also observed significant differences between environments for wheat yield and for grain protein content. At this point, it is very important to remember that because these fields are considerably large, not all observed variation that we can see here can be explained only by the lentil or the wheat genotype, because soil heterogeneity across the field can also influence the crop performance, and if we do not account for it, could be mask the true genetic effect. So let's talk a little bit about spatial heterogeneity. And here we can see one of these sites where the rotation trial was planted between 2024 and 2025. And if we map the elevation across the field, we can see a clear gradient where one corner at the bottom of the field is higher, at the bottom is below than the other, is lower than the other. And this can affect the crop performance. For example, the lower part of the field may have more water availability, and lentils or wheat in that area may yield more independently of the genotype used. So after feeding the mixed linear models that account for the row and column structure of the experimental design, we can estimate the spatial trend for each trait and site. And when we represent this spatial trend as a heat map, for example, for the lentil yield in 2024 at the top and wheat yield 2025 at the bottom, the patterns actually are very similar. And we can see that the highest yields appear in the lower elevation corner, probably because of this greater water availability I mentioned. But this is not all, because in addition to correcting for soil heterogeneity, we need to account for other components when estimating this legacy effect of each lentil genotype on a specific wheat trait. So the model that I'm showing here, that can look a little bit intimidating at first, but each term has a clear meaning, and I'm going to explain it to you. The first term observed is the value for a specific wheat trait in the year 2 of the rotation. The next one, mu, is the average value of that trait within each environment. The next term is actually the effect of the previous lentil genotype grown in that plot on year 1. And actually, this is the predecessor effect of interest, and it is the component that we want to isolate as the legacy effect. The model also includes the replicate of each lentil-wheat combination, a correction for the wheat genotypes used in the year 2, spatial correction based on rows and columns within each environment, and also epsilon, the last one, is the residual error. And as I said, the key component for this presentation is the legacy effect on the observed wheat trait. So let's continue with that one. And as you can see here, you can see there is variability in the legacy effect on the yield of the following wheat crop. So this is the legacy effect of the lentils for the yield of the wheat crop. And because this rotation cycle was replicated in 2 locations, you can see the results of one location at the top and the other one at the bottom. We can also evaluate whether the legacy effect is consistent across environments. So if we represent the same results, but in a scatter plot, some of the lentil genotypes show has a positive effect on wheat yield in both environments, that are those in blue, suggesting that they may leave a better condition for the following wheat crop. And at the same time, in the red dots, the lentil genotypes can show a negative legacy effect in both locations. We have not fully quantified all the genotype by environmental interaction yet, but it is important to remember that 2024 and 2025 environment were quite different. And because of this, the interaction between legacy effect and environment may be influencing these results. As I said, this is the legacy effect on yield, on wheat yield. And the next one, we can do the same analysis for the wheat grain protein content. So this is the legacy effect of the previous lentil on the grain protein content. And again, we can observe the variation within this lentil legacy effect. And again, some lentil genotypes are consistently improve the protein content in the following wheat crop across both environments, while others show a negative effect. And this suggests that the lentil legacy effect is not only limited to wheat yield, because it may also influence the wheat grain quality traits, such as in this case, the protein content. Okay, so now we have observed that this lentil legacy effect actually exists, and it can improve or reduce the agronomic performance of the following wheat crop. The next important question is, okay, so what drives this effect? So here I show you some examples where specific lentil traits were significantly associated with the lentil legacy effect on the wheat yield. So on the left, we see the relationship between lentil seed protein content and the lentil legacy effect. In one of the two sides, this relationship was negative and significant. And the site was in Clavet in 2025. At the same time, the lentil legacy effect was also negatively associated with the nitrogen harvest index. This index is important because it represents the relationship between the nitrogen allocated to the seed and the nitrogen allocated to the remaining biomass. So this suggests that when more nitrogen is exported from the system through the lentil seed, less nitrogen may remain available to benefit the following wheat yield. However, when we look at the hunter side, the trait with the strongest association with the lentil legacy effect on wheat yield, this time was related to the diversity of the microbial community living in the lentil soil. So this is especially interesting because it suggests that the effect is not only direct through lentil plant traits itself, but also indirect. Because the lentil genotype may influence the soil microbial community, and a more diverse microbial community may then have a positive effect on the yield of the following wheat crop. So in my opinion, this is one of the most exciting results so far because it connects the plant genetics, the soil biology, and the crop performance in the rotations. So for the lentil legacy effect, we saw the lentil legacy effect on wheat yield. Let's see what are the drivers of the lentil legacy effect on the wheat grain protein, because the pattern this time is quite different. In this case, the legacy effect was more strongly associated with traits such as total lentil yield and lentil seed protein content. And actually, this makes biological sense because the nitrogen allocated to the lentil seed protein is removed from the rotation system at harvest. So therefore, the lentil genotypes that produced higher seed yield or higher seed protein may export more nitrogen from the system and potentially reducing the amount of nitrogen available to the following wheat crop. So to finish this results section, I would like to show one simple way to visualize whether the previous lentil genotype can influence the following wheat crop. And for this, it's necessary to bring back the experimental layout. So if this is the plot layout in the field, within each block planted in the year 1, we included the same 3 lentil control genotypes. And in the following year, just one wheat control genotype was planted on top of these 3 lentil controls. So moving from one slide to the other, you can see the differences as we planted first the 3 lentil checks, and then on top of those 3, the same exact wheat genotype. So this is important because this gives us 96 replication for each previous lentil control genotype. So when we compare the wheat control grown after the 3 different lentil controls, we can find a significant difference between lentil 1 and lentil 3. And on average, the wheat grown after the lentil 3 yielded around 2% more than the wheat grown after the lentil 1. It is true that a 2% difference may sound like a very small effect. However, if we consider that Canada produced more than 40 million tons of wheat in 2025, a 2% increase in production would represent almost 1 million additional tons of wheat. And this simple example shows why even a small rotation effect can be agronomically and also economically important when applied at a large scale. So to conclude this presentation, I would like to first emphasize that the results I have shown today came from the first complete rotation cycle, and this cycle includes 4 replications for each lentil-wheat combination. Once we have completed the next rotation cycle, which will finish this season, because in this season we will plant the wheat again on top of the lentils that was planted last year, we will have 4 additional replications in new environment, which will make the results more robust. So the main take-home messages from this trial are the following. First, we have identified a measurable legacy effect on the lentil for the following wheat crop. The second, this legacy effect looks that can be influenced by the environment. The third one, the lentil legacy effect can influence both the wheat yield and wheat grain protein content. This effect seems to be driven by direct lentil traits, such as the nitrogen allocation within the plant. And also, finally, it also seems to be influenced by indirect plant effects, including the soil microbial diversity associated with different lentil genotypes. So, finally, the next steps will be to integrate all the data layers and replications across environment, compare the different statistical models and select the most efficient ones, and also identifying, of course, the genomic regions associated with this legacy effect, and test whether a true genotype by genotype rotation effect can be detected between the lentil and wheat crop. Taking into account, of course, that identifying these genomic regions associated with the legacy effect and to the rotational value would also allow us to predict new phenotypes without the need to plant this giant experiment in the field again. So finally, I would like to acknowledge to the founders, because this project has been possible thanks to many people and institutions. And first, I would like to thank to the funding agencies that supported this work. And of course, I also like to thank to the project team, all the collaborators, field and technical staff, and students to make all this project possible. So thank you to all. This was the last slide, and I would be happy to answer any question you may have.
Thanks, Salvador. That was a very interesting and quite impressive presentation. So
Thank you.
let's see if there are any questions from the audience. You can just unmute and talk. Tom, I see you raising your hand.
Yeah, hello. Thank you so much, Salvador. That was wonderful presentation. I want to follow up a little bit on your legacy effect story, and whether you have already or you will in the future think about like which types of lentil seem to have the better legacy effect for wheat in terms of maybe their phenological characteristics, like the taller ones or the later maturing or higher biomass. Do you have any clues about that already?
Well, actually, it is true that maybe the legacy effect is not very connected to the phenology, but it is connected to other traits, like, for example, the nitrogen content in the biomass. And actually, this makes sense, no? Because if there is more nitrogen in the biomass, this would be more nitrogen in the lentil residue, and that would be more nitrogen in the rotation system for the following crop. So actually, the thing is that we have seen that the lentils that yield the most are the lentils that leave less benefits for the following crop. And actually, this also makes sense. But the thing here is that we wanted to identify the genomic signatures in the genome for make the lentil also better in the rotation without including this trade-off in the nitrogen allocation.
Thank you.
And actually, I would like to say also that this is the model we have used for the project, lentil and then wheat. We have been using this rotation system as model in the project because it's what it works here in Canada. No, it's a rotation that the farmers commonly use. But after identifying genomic regions associated with this legacy effect, would be also super interesting to see if we can export this to other crops, like, for example, I don't know, peas and oat or different pulses.
Okay, I see another hand. So please speak up. I only seen the notification.
Oh, I think you are mute.
Azim, you are mute.
No.
Okay, now.
Okay.
Sorry. So here you have studied the effect of different lentil genotypes that affect the wheat yield. So here the nitrogen fixation, biological nitrogen fixation of lentil genotype may be vary. So the first thing is that biological nitrogen fixation B and F of lentil genotypes, if you have studied it at first, then it will be much more clear that this genotype has much more BNF ratio that will affect the wheat yield. Is it?
I'm not sure if I got the entire question. Can you say that again a little bit louder? Sorry.
Aziman, sorry, your audio comes a bit disturbed. So
Yeah.
please, if you could speak slower, or you can type in the chat if it's not
Okay,
possible. Sorry.
okay, okay, not matter. I am saying that the lentil genotypes you
Yeah.
have used, the BNF, biological nitrogen fixation rate, is the same there because different genotypes have different biological nitrogen fixation rate.
Yeah. Okay. I got it.
That, that that will affect the next crop which will be going to sow, whether it's wheat, whatever. So the BNF ratio, the BNF rate is the same in lentil genotype which you have used. Is it?
Well, yeah, thank you for the question. It's a really good one. The thing is that I would like to say that this field experiment is within a larger project. So in this project, we have different activities now, and other people are also looking at these kind of things. For example, we have a grad student working on the BNF, how the difference is in biological nitrogen fixation, and how this could also modify the agronomic performance of the following wheat. For example, that one is super interesting because what that student is doing is to incorporate N15, that's using as a traceable nitrogen label. And he's putting the nitrogen 15 first, growing the collection of lentils, and then growing the same wheat genotype over on top of those residues, not those previous residues. So you can trace exactly the nitrogen movement from one crop to the other. So stay tuned because we will have those results very soon. And would be nice, yeah, to show also this variability from the lentils perspective from the BNF.
Okay. Thank you so much. Thank you.
Yeah, actually, the thing is that from a field experiment, we cannot, or it's more hard, to take a look at the BNF because we need to harvest also the roots, maybe counting the nodules and everything. So this is something that would be more feasible in a more controlled experiment in a greenhouse growth chambers or something. So this is what we are exactly doing.
Thank you.
Okay. Are there any other questions from the audience?
Can you hear me?
Yes.
Yep, I can hear you.
Yeah, I'm Alain Baranger from France. Very, very nice talk. Really, really very interesting. Thanks a lot.
Thank you.
I was wondering about dissecting direct and indirect effects of lentils. Do you have any idea if these direct and indirect effects interact positively or negatively, and if this would have an effect on breeding schemes, for instance? If you try to breed for direct effects, would you have negative indirect effects, or would you have also positive indirect effects? Am I clear?
Yep. Thank you for the question. That's also a very interesting one because at the beginning I wasn't sure if calling direct and indirect effect. I was calling direct effect to the, for example, to the effect of the legacy effect if it is a direct effect of a plant trait. For example, more nitrogen content in the biomass, I call that a direct effect. And I call an indirect effect if, for example, the lentil is shaping the microbiome, the microbial communities diversity in the soil. I call that an indirect effect because it's not directly related to a plant trait.
Yeah.
I'm not sure if I explained myself very good. So in this case, we saw that in one of the sites, the trait that modified the legacy effect the most was indeed the microbial diversity in the soil, and that was shaped by the previous lentil. So in this case, the relationship was positive, and it was an indirect effect. And
You
the
mean that
traits
the
I
level
called
of nitrogen left by the lentil would also generate a more microbial diversity?
in the case I just said was the
Okay.
the second one, the microbial diversity in the soil. But for example, if the lentil export more nitrogen to the seeds, that nitrogen would be out of the rotation system. And for example, the lentils that put more nitrogen in the seed, and the legacy effect is negative. So lentils with more protein content, the legacy effect of those lentils would be lower than the lentils that have a lower nitrogen protein content in the seed.
Okay, thanks.
Thank you.
Great. Are there any other questions? I'm just taking a look at the chat, but more or less it seems that we are done. So I'm thanking again Salvador for your precious contribution. I remind to everybody that we have our upcoming conference, which is at this point next month from 8 to 12 of June in Dubrovnik. That will be our 5th conference. All the information are in the usual places on the website. There is a dedicated website. And see you there. I know that Salvador, you will also be at the conference. And I hope to see all of you from the audience around there. Thanks again, and see you next round.
Thank you very much. See you in Dubrovnik.